Assalamu alaikum dear students, welcome to Dr. Roomi lectures. In our previous session, we studied the physiological actions of insulin. In today's session, we will study the actions of glucagon hormone, which in general opposes the actions of insulin. Glucagon is a peptide hormone which is secreted from the alpha cells of pancreas, and these alpha cells are located in the periphery of the islets of Langerhans. Glucagon hormone contains 29 amino acids and its half-life is very short, just 3 to 6 minutes. Let's talk about the mechanism of action of glucagon hormone. This is a protein hormone, easily water soluble. It finds its receptors on the cell membrane because it cannot cross the cell membranes being water soluble. On its receptors, it acts by cyclic AMP mechanism, which means that when it binds to its receptors, the second messenger system which is activated inside the cell is cyclic AMP, which is increased. Increase in cyclic AMP activates protein kinase A, which causes phosphorylation, because of which some enzymes are activated and some enzymes are inhibited. In this way, glucagon performs actions. Here it's important to note that the principal site where glucagon acts is liver. As compared to insulin, which also acts on skeletal muscles, glucagon hormone has principal actions on liver and some actions on fat cells. Glucagon hormone acts on liver to increase glycogenolysis, which means this hormone increases the breakdown of glycogen to release glucose into the blood circulation. Glucagon stimulates the enzyme which is called as glycogen phosphorylase. This enzyme converts glycogen into glucose 1-phosphate and then glucose 6-phosphate is formed. Another enzyme which is called as glucose 6-phosphatase is present in liver but not in muscle, that causes the release of glucose into the blood circulation. Liver is generous because it can break glycogen to release glucose into the blood circulation so that other tissues, especially brain, can use this glucose during fasting. At the same time, another enzyme, glycogen synthetase, which causes formation of glycogen, is inhibited. So glycogen breakdown is increased and glycogen synthesis is decreased. Glucagon increases blood glucose level not only by breaking glycogen but also by increasing gluconeogenesis. Gluconeogenesis means increase in synthesis of new glucose, and this is done by increasing the conversion of pyruvate towards glucose. Glucagon promotes the conversion of pyruvate into phosphoenol pyruvate, and then the reactions towards the backward direction, which increase the formation of glucose, are increased. Glucagon decreases the utilization of fructose 1,6-bisphosphate in glycolysis, so glycolysis is decreased, and at the same time conversion of fructose 1,6-bisphosphate into fructose 6-phosphate is increased. This fructose 6-phosphate is then converted into glucose 6-phosphate, which by the action of glucose 6-phosphatase enzyme enters into the blood circulation. Glucagon hormone also increases insulin secretion just like duodenal hormone secretin. Glucagon also increases lipolysis. A little bit lipolysis is increased in the fat tissue, but most of this lipolysis is done in liver. So it means glucagon hormone has less effect to break lipids in the adipose tissue, but more so in the liver. So as compared to cortisol, glucagon is not a major fat mobilizing hormone. Anyways, due to the lipolytic action of glucagon hormone, fatty acid and glycerol level will increase in our blood. Glycerol can be used as a substrate for gluconeogenesis in liver. Glucagon hormone also increases the beta oxidation of fatty acids in liver. It speeds glucose to be used as a source of energy. The acetyl coenzyme A, which is formed from the breakdown of fatty acids in liver, they are too much and excess of that is converted into ketone bodies. Some important ketone bodies which are released into blood circulation are acetoacetate and beta hydroxybutyrate. They may be given to muscles so that they can be used as a source of energy. These are acidic bodies, so that's why they're also called as ketoacids. Glucagon not only increases the breakdown of fatty acids and convert them into ketone bodies, which means it increases ketogenesis, but also at the same time, glucagon hormone decreases the synthesis of fatty acids. And this is done by decreasing the reaction in which acetyl coenzyme A is converted into malonyl coenzyme A. The enzyme required for that is acetyl coenzyme A carboxylase, which is inhibited by the action of glucagon. When this rate limiting enzyme for the de novo synthesis of fatty acid is inhibited, so there is less synthesis of fatty acids and hence the synthesis of triglycerides is decreased. So in a nutshell, we can say that glucagon hormone increases lipolysis, especially in liver. It increases the utilization of fatty acids as a source of energy, so it increases oxidation of fatty acids. it increases the formation of ketone bodies, so it increases ketogenesis, and at the same time it decreases lipogenesis, and also it decreases the fatty acid synthesis. Glucagon hormone also increases the protein breakdown in liver and decreases protein synthesis. Because glucagon increases gluconeogenesis, and hence the conversion of pyruvate into glucose is increased. Because of this, indirectly there is increase in conversion of amino acids towards pyruvate. For example, conversion of alanine into pyruvate by the action of alanine transaminase enzyme will be increased. Because in this transamination reaction there is a removal of amino group from the alanine which is converted into pyruvate. That amino group will be incorporated into urea synthesis. So in this way, because glucagon increases the entry of amino acids into liver, those amino acids they are utilized. For example, they are transaminated and the amino groups are removed. Those amino groups they are used for formation of urea, and also amino acids are converted into pyruvate and then into glucose. So in this way there is increase in glucose synthesis which is called as gluconeogenesis. So protein synthesis is decreased and the protein breakdown in our liver will be increased by the action of glucagon hormone. So in a nutshell we can say that glucagon has net anti-anabolic action in liver, which means in the liver it decreases the protein synthesis, and also because it increases the utilization of amino acids for formation of new glucose. So the removal of amino groups increases the urea formation, so it increases ureagenesis. What is the net effect of glucagon on various nutrients in our blood? It increases glucose because it increases glycogenolysis and gluconeogenesis. It decreases amino acids because it increases the entry of amino acids into the cells and conversion of those amino acids into new glucose, gluconeogenesis. It also increases the free fatty acid levels in our blood because it increases lipolysis, especially in liver. And it increases the formation of ketone bodies because it converts fatty acids into acetyl coenzyme A, which then are conjugated to make ketone bodies like beta hydroxybutyric acid and acetoacetate. So glucagon favors fats to be used as a source of energy, but it spares blood glucose level so that glucose can be given to our brain during states of fasting. Glucagon also has some non-metabolic actions. For example, it increases the force of contraction of heart. It also increases blood flow to the muscles. It increases bile acid secretion and decreases the gastric acid secretion. Is there any clinical abnormality which is caused by the excess or deficiency of glucagon hormone? Till now, research has not shown any specific disease which is caused by the increase or decrease in the glucagon level. However, diabetes mellitus is frequently accompanied by increase in glucagon secretion. This increase in glucagon level in diabetic patients increases blood glucose level and further intensifies hyperglycemia. The possible reason behind is that because insulin is required for the entry of glucose into the alpha cells of pancreas. When there is no insulin or less action of insulin in diabetic patients, the entry of glucose into the alpha cells is decreased. Hence, the release of glucagon hormone will be increased. For this reason, some diabetic patients who are insulin dependent are best treated not only by giving insulin but also somatostatin at the same time. Somatostatin, as you know, that is an inhibitory hormone which decreases the secretion of glucagon. So when glucagon secretion in diabetic patients is decreased, this decreases blood glucose level and improves blood glucose control. Now let's talk about the control of glucagon secretion. Glucagon hormone is secreted from the alpha cells of pancreas. Initially, when it is synthesized, it is pro-glucagon stored in the vesicles, and before its release, it's converted into glucagon and then released. Most important stimulus to release glucagon from these cells is hypoglycemia. When there is less entry of glucose into the alpha cells of pancreas, there will be less metabolism inside, and that will stimulate the release of glucagon hormone from these vesicles by exocytosis. And when there is hyperglycemia, this is most important inhibitor of glucagon release. Amino acids, especially arginine, lysine, and alanine, also increase the release of glucagon from the alpha cells. And some important hormones which are involved in increasing the release of glucagon hormone after taking a protein meal are cholecystokinin and gastrin hormones. Increase in blood amino acids level, for example, after taking a protein meal, increasing the secretion of both insulin and glucagon. This seems a very funny story. Why it is so? Let's suppose when we take only a protein rich meal and no carbohydrate. For example, if we just eat some barbecue and no rice or no bread, then what happens? There is increase in the amino acid level in our blood after digestion of the proteins. In response to these amino acids, the insulin concentration increases. And if there is no carbohydrate or very little carbohydrate, the blood glucose level will not increase, but insulin will decrease your blood glucose level. So in this way, insulin can cause hypoglycemia. So this is why it is important that the glucagon hormone in response to the amino acid is also increased. This glucagon then acts on the liver. Then by glycogenolysis and gluconeogenesis, it increases the blood glucose level. And in this way, it prevents hypoglycemia in response to a protein-rich meal. Somatostatin hormone also decreases glucagon secretion. Free fatty acids and ketone bodies, they also inhibit further secretion of glucagon. And fasting and exercise, because in response to both of these there is decrease in blood glucose level, so that low blood glucose level increases secretion of glucagon. Cholecystokinin and gastrin hormone, which may be released in response to a protein meal, they also increase the secretion of glucagon hormone. What about the action of insulin on the release of glucagon hormone? Directly, it has a parakine action on the alpha cells of pancreas, and it inhibits the release of glucagon from here. So just to summarize, glucagon is a peptide hormone which is released from the alpha cells of pancreas in response to mainly hypoglycemia and also amino acids, especially arginine and lysine. Glucagon has actions which are opposite to insulin. It acts mainly on liver and not on skeletal muscles. Insulin has its actions on muscles, adipose tissue, and liver as well. Glucagon is a hormone of fasting state. It's increased in between the meals, and it causes increase in blood glucose level by increasing glycogenolysis and gluconeogenesis. Liver is generous because it increases the release of glucose, and this is because some enzymes like glycogen phosphorylase and glucose 6-phosphatase, which are present in liver, they are stimulated by the action of glucagon hormone. Glucagon also increases lipolysis in the liver, and it decreases lipid synthesis. It also increases the use of fatty acids for beta oxidation and increases ketogenesis. It also increases the entry of amino acids into the liver cells and hence their use for gluconeogenesis. And urea formation is also increased. And protein synthesis in our liver is decreased. If you compare the actions of insulin and glucagon, insulin increases glycolysis, which means it increases the use of glucose for oxidation. It also increases glycogenesis or glycogen synthesis and increases lipid synthesis and protein synthesis. But glucagon hormone decreases glycolysis, it also decreases lipid synthesis, decreases glycogen synthesis, decreases protein synthesis. Insulin hormone decreases glycogenolysis. However, glucagon hormone increases glycogen breakdown. Insulin also decreases gluconeogenesis. However, glucagon hormone increases the conversion of glycerol and amino acids into glucose. So gluconeogenesis is increased by the action of glucagon hormone. Glucagon also increases lipolysis or lipid breakdown, but insulin hormone decreases lipolysis. Glucagon also causes the conversion of fatty acids into ketone body, hence glucagon increases ketogenesis, but insulin decreases ketogenesis. Another important action of glucagon hormone is that it increases ureagenesis, but insulin decreases ureagenesis. So we can say that insulin is mainly an anabolic hormone, but glucagon is a catabolic hormone. In our next session, we will study the regulation of blood glucose level and clinical features of diabetes mellitus. Please don't forget to like and share the video. Thank you for watching. See you next time with another video.